Hello everyone, welcome you all to my YouTube channel Let's Do Biology. I hope you all are doing well. If you are new to my channel then please subscribe my YouTube channel Let's Do Biology, and if you are liking my video then hit the like button and share it with your friends. In previous two videos I shared my interview experience of IISc Banglore. So in this video I am gonna share my interview experience of National Institute of Immunology, Delhi. Okay, so myself Archana, I am research scholar at IISc Bangalore. So let's start now. आज के वीडियो में मैं अपना NII दिल्ली का PhD इंटरव्यू एक्सपीरियंस शेयर करने वाली हूँ कि किस तरह से मैंने फर्स्ट राउंड ऑफ इंटरव्यू में क्वेश्चन मिले मेरे को एंड हाउ आई आंसर्ड दैट questions, and in later in second round of interview, in that how I get my questions, and same again how I answered those questions also, so that you will get some idea that which type of questions you can get in your interview. So first of all, NII में दो राउंड ऑफ इंटरव्यू होता है, फर्स्ट राउंड एंड व्हेन यू क्लियर फर्स्ट राउंड देन लेटर ऑन यू विल अपीयर फॉर सेकंड राउंड ऑफ इंटरव्यू. ठीक है, उससे भी पहले आपको NII का अलग से एग्जाम क्लियर करना पड़ता है. NII का अलग से फॉर्म निकलता है, वो फिल करना पड़ता है, देन आपको एग्जाम में अपीयर होना पड़ता है. इफ यू क्वालिफाई दैट एग्जाम देन यू विल कॉल्ड फॉर फर्स्ट राउंड ऑफ इंटरव्यू. ओके, सो ठीक है, with this information let's start now. So आपको like it was my first interview was around in June, like near to CSIR net exam. Okay, so what question was asked in my first round of interview? So there were like around 6 to 7 professors were there in my panel for first round of interview. So I think three professors were there which asked question to me. सो दे क्वेश्चंस आर लाइक, इसका मेरे को इतना ज़्यादा लाइक क्वेश्चन याद तो नहीं था, बट जितना याद था आई ट्राई टू फ्रेम दोज क्वेश्चंस. ओके, आई थिंक दिस विल ऑल्सो हेल्प टू यू टू गेट सम आइडिया ऑफ इंटरव्यू ऑफ NII, क्योंकि मेरे को कहीं भी NII के इंटरव्यू का कुछ भी नहीं मिला था. सो आई थॉट कि it will help you. Okay, so the first question which asked to me in interview was how so first they ask you your interest of topic. So again I gave my interest of topic as cell biology and immunology. So as it is focused on immunology and I is just focused mainly on immunology, so they asked me question, I think mostly on immunology only. ठीक है, सो जो फर्स्ट क्वेश्चन उन्होंने पूछा था विच वाज हाउ टी सेल इन्वॉल्व्स इन इम्यून रिस्पॉन्स. ओके, सो दे वर आस्किंग कि टेल दी लाइक कुछ भी इम्यून सिस्टम का ऐसा पाथवे लाइक या ऐसा कुछ भी रिस्पॉन्स जहाँ पे टी सेल इन्वॉल्व होता है, तो आपको वो सब बताना था कि किस तरह से टी सेल एंटीजन प्रेजेंटेशन लाइक एंड रिस्पॉन्स में कैसे इन्वॉल्व होता है, कैसे साइटोकाइंस एंड ऑल, ये सब बताना था सबसे पहले जो क्वेश्चन पूछा था उन्होंने. ठीक है, so I gave the answer of this question very well. Then further they asked me tell us different type of as I included APCs in my first question. So further they asked ki, do you know about different type of APCs? If you know then tell us the name. Okay, then I just tell the name of like B cell, macrophages, dendritic cells. Okay, and they were like okay. Then further they asked me intracellular pathogen, like intracellular pathogen activate, there are two pathway for MHC na, MHC1 or MHC2. So they were asking ki intracellular pathogen activate which MHC. Okay, so this मेरे को अच्छे से तो क्वेश्चन याद नहीं, बट जितना याद था आई ट्राई टू फ्रेम दैट. ठीक है, इंट्रासेलुलर पैथोजन जो एक्टिवेट करता है MHC को. सो इट वाज लाइक MHC1 ना, वायरस जो होते हैं वो सब MHC1 को एक्टिवेट करते हैं. सो आई इंक्लूडेड CD8 प्लस इन माई क्वेश्चन in previous this question MHC wala question. So further they just frame the question on that word कि CD8 plus involves in which pathway. Okay, so I just said in MHC1, and also explain the pathway. Then further the question was, if T cell not secrete cytokines, then how B cell activates and make antibodies. Okay. Then further they are, I was little bit confused in this question, but after lit, uh, after I was able to answer it this also, they give me some hint related to this question. Okay, so further what they ask, different type of T cell, like they were asking name the different type of T cells, and they will differentiate in different types. Yeah. So I need to tell the name of the different type of T cells. Like we know that there are like six to they differentiate in like 6 to 7 different types of T cells. So I gave the name of all type of T cell. Okay, and then I included Treg in my in this question. ठीक है. So further what they frame the question on that word only. So जब भी आप कोई भी question का आंसर दो, तो सोच समझ के देना कि आप जो भी उसमें वर्ड बोल रहे हो, यू आर फेमिलियर टू दैट. अदरवाइज वो आपको उसी से रिलेटेड क्वेश्चन पूछते हैं, और अगर आपको नहीं पता होगा तो उसका आप गलत इंप्रेशन पड़ता है. ठीक है, तो वही वर्ड बोलना सोच समझ के जिसका आपको नॉलेज हो. ठीक है. सो दे आस्क मी व्हाट आर टी रेग सेल, देन आई गिव द आंसर दैट व्हाट देयर फंक्शन एंड how they regulate our immune system and what are their marker and all. Okay, so I gave the I include foxo p1 in my answer, foxo 1. ठीक है. Then further what they asked, how we determine or isolate the Treg cell, then I also gave the answer of this question on the basis of that transcription factor which is particular for Treg cell. And then I also explained the technique that how can we isolate. ठीक है. Then you also need to know about the technique that includes in particular topic which you are preparing for your interview. So further, I think this was asked by two professor, and then further one more professor asked me these questions. But they were, how can we determine the level or amount of mRNA? Okay, mRNA का level कैसे determine कर सकते हैं? Then I was little bit confused. I don't know why, but later I gave the answer that by RT-PCR, northern blotting, we can determine this. And she was like, okay. Then further she asked me, how can we determine that the protein is formed by that mRNA, जो हमारा लास्ट में इसी क्वेश्चन से रिलेटेड किया कि mRNA फॉर्म हो गया और इससे हमारा प्रोटीन फॉर्म हो रहा है या नहीं, हम वो कैसे डिटरमाइन कर सकते हैं. देन अगेन आफ्टर बिट ऑफ टाइम आई गेव द आंसर इज सदर्न ब्लोटिंग. सो शी वाज satisfied with this answer. And then further she framed one more question, that if I have mRNA of TNF alpha, IL-2 and beta M, I mean beta microglobulin, but I am not getting band of the protein by doing western blotting, what can be the reason? Okay, although I was not able to give the answer of this question at that time, then I just said, ma'am, I am not getting the answer right now. So she said like, okay. And this was for my first level of interview. After like uh, my first interview was on 8th, and then after 2 days, I think on 10th I got the mail that I am selected for second round of interview. And then later I appeared for second round of interview, it was on 12th of June. Okay, ठीक है. So this much span of time you will get after first round of interview to appear in second round of interview. So further the question, second round of interview का मेरा इतना ज़्यादा लंबा नहीं गया. फर्स्ट राउंड कंपैरेटिवली लंबा गया था, क्योंकि सेकंड राउंड में जो प्रोफेसर थे वो काफ़ी जल्दी में थे, उनका काफ़ी टाइम हो चुका था उस टाइम तक, तो वो ज़्यादा क्वेश्चन नहीं पूछे. एंड ऑल्सो मेरे को सेकंड लेवल का इतना ज़्यादा अच्छे से याद भी नहीं है. ठीक है. सो जैसे अगेन दे आस्किंग व्हाट इज योर टॉपिक ऑफ इंटरेस्ट, एंड देन आई अगेन टोल्ड कि सेल बायोलॉजी एंड इम्यूनोलॉजी, एंड दे फ्रेम्ड द क्वेश्चन फ्रॉम सेल बायोलॉजी, सॉरी इम्यूनोलॉजी. वन प्रोफेसर आस्क्ड, मतलब जैसे इन इट वाज लाइक NII का ऑनलाइन इंटरव्यू होता है. ठीक है. सो सबसे पहले उन्होंने डिजाइन एक्सपेरिमेंट ही पूछ लिया, एंड आई वाज ब्लैंक. I don't know what happened to me, but I was blank. First question was design an experiment that B cell originate from spleen or transplantation, something like that. That professor asked me. I think this question is not framed well, but still I just wrote what I remembered. क्योंकि मैंने क्वेश्चन काफी टाइम बाद लिखा था मेरे इंटरव्यू के, सो इतना अच्छे से याद नहीं था, तो okay, please bear me for this. Then the next question was, tell us the different type of cell blood present in blood. And then they were asking like WBC, RBC, platelets, and all. And then further they asked me which cell carry oxygen. And I said like RBC. Then they asked further asked me the question related to RBC only, why RBC is enucleated and what is its shape. Then I also explained this question. And further the question was related to earlier questions, that why if RBC carrying oxygen, then why WBC can't carry oxygen. Okay, the question is something like that. Then further they asked me precursor of RBC and related to these type of questions. And further मेरे को इसका और क्वेश्चन अच्छे से याद नहीं है, बट आई थिंक एक दो मोर क्वेश्चन होगा, बट रिलेटिवली ये मेरा सेकेंड राउंड ऑफ इंटरव्यू इतना ज्यादा लंबा नहीं गया, ठीक है. Then further, I get selected in second round. I think final round of interview, and the list was like this, and it comes on 15th. My second round of interview was on 12th of June, and the list appeared on came on 15th of June. After 3 days, okay, and I got selected in NII second round of interview. Okay, so I hope guys this will help you further in your guidance. Thank you. If you are liking my videos, then guys please share it with your friends so that they can also get benefit like you are getting. So, and also I will get motivated from your. Okay, thank you guys for watching my videos. Please subscribe my channel, let's do biology. And if you are new to my channel, then again guys, subscribe my channel and share it with your friends. Thank you. We'll meet you in next video. Till then, take care.